So the artwork that I was working on this morning worked out really well. That's it. Finished. So I'm gonna finish talking about the plant sciences great course that I was. That's sort of sideways. We'll see. I should actually set that up on a tripod or something like that so it's not crooked, but I never do. So, I was talking about it trying to interest people, and it turns out that I interest Japes in it because now he's reading the book, and he went through a couple of the lectures already of the plant sciences and introduction to botany. So, I'm going to talk about lecture 24. Corn pollen travels far, and a single cob has only one mother but could have several fathers from all over the place. In grasses, seed and fruit are fused into a single caryopsis. Silks are leftover stigmas where pollen grains landed to fertilize each fruit kernel. One cob can be variegated because of this. Corn didn't seem to follow Mendelian pattern. Homologous chromosomes are same size and shape, one from mum, one from dad. Process happens during creation of sex cells, sperm and eggs. Cornell Barbara McClintock used corn cell squashing by slicing sections of corn into very thin sections and then squashing them between slide and cover slip. The chromosomes in the cell became visible. She was able to ID chromosomes in corn, all 10. She noted knobs on the ends of chromosomes. She was the first to observe the process of crossing over when viewing chromosomes with the microscope. McClintock was interested in the variegation of corn. The patterns of variegation couldn't be predicted. Some corn plants had a fair number of broken chromosomes. These broken chromosomes were heritable. By observing where a chromosome was broken and what physical characteristic went with it, she could determine what a particular gene should have been doing. Broken chromosomes linked with pattern of variegation. When chromosomes broke, pieces of DNA moved and reinserted themselves in another part of the chromosome. She called the broken pieces controllable elements. She won a Nobel Prize for her discovery of mobile genetic elements, which are now called transposons, or jumping genes. They create new combos in the genome, which lead to adaptations. Nature uses gene jumping. Bacteria and viruses can and do facilitate gene jumping across species. Humans have been creating genetically modified organisms. They transfer a piece of DNA from one organism into another organism. The genome of an organism is the combo of all the DNA. A little DNA from one organism into another modifies it. Bt, Bacillus thuringiensis, live naturally in the soil. Bt produces a proton that can ward off a major corn pest, the European corn borer, a type of moth. The larval stage eats corn leaves and stems. Most bacteria produce endotoxins. One gene produces Bt endotoxin. Inserting only one gene into corn made corn resistant to the European corn borer. There is now Bt cotton, Bt potatoes, Bt eggplant, and Bt soybeans. Switzerland banned the use of Bt crops. Corn borers and other lepidopterans are just trying to live. If they happen to develop a resistance to Bt toxin, they'll thrive while others die off. Pesticide resistance develops no matter where the pesticide comes from. The Bt gene is inserted with the assistance from another bacteria. Bacteria have plasmids, unlike us, small circular pieces of DNA that are separate from the regular chromosomes of the bacteria. Plasmids Plasmids are partially responsible for bacterial development of antibacterial resistance quickly. Plasmids can be shared between bacteria. DNA within plasmid can be sliced up using restriction enzymes. Enzymes are proteins that speed up reactions. Restriction enzymes cut DNA at precise places. You can use Control Find to search text for a word. In the same way, restriction enzymes find genes and cut them in the same location. Then when cut occurs, the ends of the DNA are now sticky. They attach to other DNA pieces. GMOs are most commonly created using Agrobacterium tumefaciens. In nature, it infects plants and causes crown gall disease, which is a tumor-like growth in the plant. It gets the plant to make this tumor-like growth by infecting it with its own bit of DNA, which is called transferred DNA. This tDNA is on a plasmid, and because the plasmid causes tumors, it's called a tumor-inducing plasmid, a TI plasmid. The plasmid of the Agrobacterium tumefaciens are called TI plasmids because they are normally tumor-inducing. The plasmid is extra. The bacteria can function normally without it. One removes the TI plasmid, cuts it with a restriction enzyme, and inserts the donor gene into the plasmid. Then one reinfects the plant host with the modified plasmid, which will insert itself into the plant's DNA, just like it would in nature. Then instead of the plant producing the tumor-like growth, the plant will produce whatever gene you inserted into the TI plasmid. In the case of Bt corn, the plant will now make the Bt endotoxin. The TI plasmid had to be modified itself, though. Otherwise, it would still cause tumors. The tumor-inducing genes are removed from the plasma and markers are added. These markers make it easy to test whether the plant has successfully taken up and integrated the modified plasmid. The marker is usually a gene that confers resistance to the antibiotic. Then you try to grow the bacteria in the presence of the antibiotic. If it doesn't grow, then it didn't pick up the plasmid. If it does grow, then it successfully picked up the gene of interest and the test marker, which tested positive for growth in the presence of antibiotic. This was how genetic engineering got started. It can still be done this way, but now there are actual gene guns. These use nanometer-sized small gold or tungsten bullets that are coated with the DNA of interest to insert it into the plant's DNA. First invented in the mid-1980s, the gene gun technology was the way Roundup-ready soybeans were created. Glyphosate will kill pretty much any plant. It shuts down an enzyme that plants need to synthesize at least 3 amino acids, which are building blocks of proteins. The enzyme is called 5-enolpyruvishikimate 3-phosphate. Look it up. EPSP synthase. When this EPSP synthase enzyme is shut down, the reaction to make the amino acid stops and the plant can't survive. Generally, glyphosate is non-toxic to vertebrates and insects. Only plants and microbes produce the EPSP synthase enzyme. Only plants and microbes will be harmed by the glyphosate, which knocks out this enzyme because glyphosate is so damaging to plants. The way Roundup Ready crops work is a bit different than the way the Bt crops work. With Bt crops, crops make their own pesticide. Plants can't produce their own glyphosate because they would inhibit their own EPSP synthase and kill themselves. Agrobacterium tumefaciens isn't killed by glyphosate. Monsanto scientists located a gene responsible for producing a different enzyme than EPSP to make essential amino acids that the plant needs. This gene, inserted into soybeans, made it so plants could be sprayed with Roundup and wouldn't die. Only the surrounding weeds will. The gene gun was used to get Roundup-ready gene into soybeans. Monsanto received a one-time payment of $450,000 in 1992 for the gene from a company, DuPont, so they basically gave away their discovery. DuPont produced Roundup Ready seeds, and Monsanto profited from the increased sales of Roundup. In 2000, the first glyphosate-resistant horseweed, Conyza canadensis, was found. It was found in the middle of a soybean field. Later, other resistant weeds were discovered. Monsanto then developed a second generation of Roundup Ready crops. Glyphosate harms an enzyme humans don't even produce. We can't even make the amino acids that the enzyme catalyzes. BT corn makes a pesticide that doesn't harm us, only caterpillars. Allergies? Superweeds? Will BT corn genes end up in other plants? There has been a problem with pollen contamination. Corn is wind pollinated and travels far. Friendly insects eat pesticide plants and die. Monarch eat milkweed near corn, but monarch decline was found to be caused by South American habitat loss instead. Genetically modified genes passed on to offspring, and farmers are forced to buy new seeds each year. Egyptian scientists developed a drought-resistant form of wheat by inserting a gene from barley. The new wheat outperformed the non-GMO variety under normal rainfall. Currently, Egypt has to import wheat. Switzerland researchers put a daffodil gene into rice to increase vitamin A content. Golden rice used a plant pigment, beta-carotene. They were going to give rice away, but were confronted with sustainability issues. Japanese Science 2007 developed new vaccine for cholera put into rice. 2 rice strains modified to carry CTB gene, which codes for a protein found in cholera. Exposure through eating causes people to develop antibodies, which then would improve their immunity to the disease. Rice mostly isn't digested in the stomach. 3/4 of the protein of interest can make it through the stomach to be absorbed through the mucous lining of the small intestine. Danielle U of Central Florida engineered plants to deliver insulin to patients with juvenile onset diabetes. Genetically engineered lettuce plants produce insulin inside their cell walls. The cell walls are made of cellulose, and they don't break down initially until good bacteria in the gut break them down. These bacteria absorb the insulin and release it into the bloodstream. 2016 German report showed that just spraying RNA on barley leaves decreased fungal infections. Scientists have observed patterns of genome of some bacteria. Some base pairs, which make up the DNA, would be repeated over and over again. In between repeated segments would be normal areas of DNA. These patterns are called clustered regularity interspaced short palindromic repeats, or CRISPR for short. These repeats match DNA of viruses that prey on the bacteria. CRISPR holds onto pieces of viral DNA, so bacteria will recognize future attacks from viruses, much like our own immune system. The bacteria with CRISPR also have a group of enzymes called CRISPR-associated proteins, or Cas for short, that will cut the DNA in precise locations. Scientists can add, delete genome genes with accuracy and speed. Now, I also wanted to mention something from another lecture. It's just quick. This is from lecture 10. They were talking about how cycads, they used to be used for food quite a lot from, like, in a lot of different cultures. And then they stopped. Like, they used to make miso, the Japanese, from cycad seeds, and now they're made from fermented soybeans. Anyway. Where is it? BMAA, beta-methylamino L-alanine, is a non-protein amino acid. Normal incidence of ALS is 1 in 50,000 people, but in the Chamorro villages, the incidence of ALS was about 200 times higher in the middle of the 20th century, 1 in 250 people. The rate has dropped to 10 times higher than normal. BMAA was a potential cause of ALS and related neurological diseases. The Camaro people ate cycad seeds and flying foxes that also eat cycad seeds. Classic case of biomagnification. The cycad has a symbiotic bacteria that lives in its roots that is the source of BMAA. The bacteria is called blue-green algae, cyanobacteria. Although it is not an algae, it's a bacteria. Blue-green algae. Wait a minute. It's a bacteria. Anyway, yeah. Blue-green algae is called spirulina and is touted as a superfood. Is blue-green algae spirulina responsible for neurological disease? Cycad conservation is required. Maybe. Only introduced animals seem to be poisoned by cycads. I don't know. Anyway. So yeah, I just wanted to mention that because somebody had asked me about blue-green algae. They said that they'd heard that it wasn't that healthy or something, or that it wasn't healthy at all. And that's what it would be, is because of that. No, I don't necessarily have an answer for you. I know Carolyn Clyer, she'd mentioned that she wouldn't eat spirulina because of that. So she was concerned about that. And others have mentioned it to me, but I don't know. I mean, sometimes if you have a little bit of something, it's very healthy, and then if you eat a whole lot of it, it's really not. Or if you eat so little, then it may not do anything. So I'm not really sure what the case is in terms of superfood or poison, blue-green algae. I don't know. So, but I thought I'd mention it. So then you're aware of it, and you know where to find it. It's in chapter 10, lecture 10. You can look that up for yourself. And then look into it further if you care. So I hope you found that interesting. I certainly found this great course great. And I'd recommend everybody goes through it. And I think James is finding it interesting too. Well, right now he's reading the book over again from The Remarkable Science of Ancient Astronomy. And we will be talking about that later.
I'm actually reading it for the first time.
Oh, you didn't? Well, we already watched it.
We watched it, but I'm reading. So.
Okay.
Yeah.
So
yeah,
there
it's
you go.
it's like 250 plus pages, but and I'm using this reading week to read.
To catch up on this stuff, because yeah, he was busy. I was doing this sort of thing, but he was doing university courses, so he got a little behind on this stuff.